Good afternoon to all the EAPCET aspirants. This is your Prasanna ma'am from NEET WORLD, and yes, in the previous session we have dealt up till prokaryotic cell from the chapter Cell the Unit of Life. And today, in today's session, we are going to begin with first the differences between prokaryotic and eukaryotic cell, and then we are going to look into plant cell and animal cell in detail, after which we will look into the limiting boundaries of a plant cell, that is cell wall, and the limiting boundary of an animal cell, that is cell membrane, after which we will move on to single membrane bound organelles and non-membrane bound organelles. So yes, let's get started with the primary differences with the in between prokaryotic cell and eukaryotic cell. This seems to be very basic, but then when assertion reason type of questions and statement based questions are asked with respect to the differences between prokaryotic and eukaryotic cell, you may make mistake. So look into it clearly, ma. First, let us go from out to in. In a prokaryotic cell, the outermost limiting boundary is what, beta? Cell envelope, right? Whereas in eukaryotic cell, if it is a plant cell, the outermost limiting boundary will be cell wall. If it is an animal cell, the outermost limiting boundary will be cell membrane. Clear, right? And then you look into the cytoplasm. Cytoplasm, both in a prokaryotic cell and eukaryotic cell, is the main arena of all the cellular activities that are taking place inside the cell. And now we will look into the non-membrane bound organelles. Non-membrane bound organelles which are common in a prokaryotic and eukaryotic cell is ribosomes. Ribosomes are common in a prokaryotic and eukaryotic cell, but here ribosomes will be of 70S type, in eukaryotes ribosomes will be of 80S type. There are two organelles which are chloroplast and mitochondria. Mitochondria is also already given here, along with mitochondria it is chloroplast. These two organelles have evolved from prokaryotic cell, which is addressed in endomembrane system. Sorry, this is addressed in what theory? Endosymbiotic theory. Okay, endosymbiotic theory. In endosymbiotic theory, we have learnt that chloroplast and mitochondria have evolved from a prokaryotic cell. That is the reason chloroplast and mitochondria will have 70S ribosomes. Okay, they will be having 70S ribosomes. Okay, and then here when you look into the membrane bound organelles, the non-membrane bound organelle along with ribosome is centrioles, which are present only in the animal cell. Okay, centrioles are the non-membrane bound organelles which are along with ribosomes present only in the animal cell. Plant cells will not have centrioles. And then if you see single membrane bound organelles which are common in plant cells and animal cells includes Golgi bodies, endoplasmic reticulum, vacuoles and lysosomes, and microbodies as well. Double membrane bound organelles in a plant cell includes mitochondria, nucleus and chloroplast. Animal cell includes mitochondria and the nucleus. Chloroplast is absent in animal cell, we know that. So these are the primary differences, ma. Now when we come to the genetic material is double stranded circular DNA in prokaryotic cell. Okay, it is double stranded circular DNA. Whereas in a eukaryotic cell you can see double stranded linear DNA. Double stranded linear DNA in a eukaryotic cell. Clear till here? These are the basic and primary differences. So I have tabulated it in a more clear way, ma. If you see in the nucleus, there is no true nucleus in a prokaryotic cell. In a eukaryotic cell, true nucleus is present with the nuclear envelope, and DNA structure is circular. It does not have any histone proteins in a prokaryotic cell. Apart from histone proteins, it consists of what, ma? Polyamines. Okay, what are these? These are positively charged. Okay, these are positively charged proteins which will help in the condensation of negatively charged DNA. And here you see in a eukaryotic cell, this is linear DNA which is associated with histone proteins. Histone proteins are positively charged. Fine, right? And cell size, if you see, it is small cell size, it is just 0.1 to 5 micrometers. Larger cell size you will see in the prokaryotic cell, that is 10 to 100 micrometers in diameter. Membrane bound organelles are absent, 10 to 100 micrometers in a eukaryotic cell, 0.1 to 5 micrometers in a prokaryotic cell. Membrane bound organelles are absent in a prokaryotic cell, present in eukaryotic cell. Okay, and cell division is through binary fission. In eukaryotic cell it is through mitosis and meiosis. And then ribosome type, if you see, 70S ribosomes in a prokaryotic cell, 80S type which is larger, and also 70S type of ribosomes in the organelles like mitochondria and chloroplast is present in eukaryotic cell. Cell wall composition, in bacteria it is peptidoglycan, which is composed of amino acids and polysaccharides. Fine, right ma? It is composed of amino acids and polysaccharides. And here cellulose is the major cell wall composition in plants, chitin in fungi, and cell wall is absent in an animal cell. Examples of prokaryotic cell, if you take, bacteria and archaea are the examples. In a eukaryotic cell, if you see, plant cells, animal cells, fungi and protists. These are the examples of eukaryotic cell. Plasma membrane will lack sterols in a prokaryotic cell. Plasma membrane contains sterols in a eukaryotic cell. Respiratory enzymes are present in cell membrane. It is because of what structures, beta? Mesosomes. We have learned, right? Mesosomes of a prokaryotic cell are equivalent to mitochondria of a eukaryotic cell. Clear, right? You are clear on that note, right? And here in mitochondria, respiratory enzymes are produced. Here in cell membrane, that is because of what structures, ma? Mesosomes. And next, if you see eukaryotic cell in detail, this is plant cell. Plant cell. Okay, in plant cell the outermost limiting boundary is what, ma? Of course it is cell wall. Clearly look into the labelings properly. Okay, look into the labelings more keenly. Fine, right? And here the cell wall will have minute pores or openings which are allowed for plasmodesmatal connections. Okay, here you can see the plasmodesmatal connections of cell wall. This plasmodesmata is the only living component that is present in the cell wall. Okay, plasmodesmata is the only living component which is present in the dead cell wall. This plasmodesmata consists of desmotubulin proteins. Okay, this consists of desmotubules. Fine, right? Which will connect, which will interact between two plant cells. And after cell wall, the next limiting boundary is what? Plasma membrane. Okay, plasma membrane is inner to the cell wall. Now we will first look into non-membrane bound organelles that are present in a plant cell. The only non-membrane bound organelles are ribosomes. These ribosomes are of two types in a plant cells. Both ATS type of ribosomes are observed in plant cells cytoplasm. ATS observed in cytoplasm of a plant cell. 70S type of ribosomes are present in, and 70S type of ribosomes are present inside the mitochondria and chloroplast. These are present inside the mitochondria and chloroplast. Clear, right? Be clear on that note, ma. And ATS type of ribosomes are present not only in the cytoplasm but also attached to endoplasmic reticulum. ATS type of ribosomes are present. Remember the locations. This is the only non-membrane bound organelles that you see in a plant cell. Apart from this, you have single membrane bound organelles. Single membrane bound organelles include what, ma? First it is endoplasmic reticulum, and then endoplasmic reticulum is of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. After which you have Golgi bodies or Golgi apparatus, and then you have vacuoles and lysosomes. These are the single membrane bound organelles. Okay, here if you see, ma, Vacuole is single membrane bound organelle and the membrane which is surrounding the vacuole is known as tonoplast. This is tonoplast. Remember, vacuole in a plant cell will occupy 90% of plant cell's volume. Okay, 90% of plant cells volume is occupied by a vacuole. Remember all the labelings properly, we can expect a diagram based question. And then about cytoskeleton. Cytoskeleton in a plant cell includes microtubules, microfilaments and intermediary filaments. But here only microtubule is highlighted. Okay, remember these are what? Microtubules. And as I told you, cytoplasm, which is the main arena of all the cellular activities. Peroxisomes, these are microbodies which are again single membrane bound organelles. Okay, remember the cycle of photorespiration where peroxisomes are involved, and peroxisomes also help in the oxidation of long chain fatty acids. Okay, peroxisomes also help in oxidation of long chain fatty acids. And then we move on to double membrane bound organelles. Of course, first it is chloroplast and then nucleus and then mitochondria. Fine, right? And here there is one more labeling inside the plant cell wall, which is middle lamella. When we are dealing with the cell wall of a plant cell, we will look into it more clearly. What is middle lamella? The first formed plant cell wall layer is middle lamella. Middle lamella is composed of calcium pectate and magnesium pectate. Okay, so here you see middle lamella is composed of calcium pectate and magnesium pectate. Remember this. Okay, this is the basic and brief description of a plant cell. Just take a note of it. Okay, take a note of it. If you are having any doubts anywhere, please drop a message in the chat box. I will be addressing your doubts. Clear, right? Any doubt anywhere, ma? Yeah. Now we will move on to animal cell. So animal cell, which this cell is from small intestine, right, where microvilli projections you can easily look into. Okay, microvilli projections are there, right, and the limiting boundary is what? Plasma membrane. Here you see non-membrane bound organelles. Non-membrane bound organelles, you will have two non-membrane bound organelles here. One is ribosomes and the second is centrioles. Okay, one is ribosome and second is centrioles. Ribosomes, same as that you have seen in a plant cell, you will have two types of ribosomes beta. One is 80S type of ribosome and the second is 70S type of ribosomes. 80S type of ribosomes is observed in cytoplasm, fine, right, and adjoining to endoplasmic reticulum. 70S type of ribosomes are present in mitochondria. Fine, clear, right? Okay, I hope you are clear on this note. Very good afternoon, ma. Continue with the class. Yeah, so here after non-membrane bound organelle ribosome, the second non-membrane bound organelle is centriole. The major role or function of the centriole is to form spindle fiber apparatus and to help in the cell division. Okay, it will look after the cell division by formation of spindle apparatus. Okay, by formation of spindle fiber apparatus or spindle fibers. It will help in the cell division. Then you might have this doubt, like spindle fibers, are they present in a plant cell? Without centrioles, how plant cells are conducting the cell division? This might be your doubt. Plant cells doesn't have centrioles, so there will not be any formation of spindle apparatus or spindle fibers. But then plant cells will have a complex called as microtubular organizing complex. Remember this term. Okay, plant cells will have microtubular organizing complex. This microtubular organizing complex will help in the formation of spindle fibers, thereby it will mediate the process of cell division. Okay, whereas in animal cells, centrioles will take care of the process of cell division. I hope this is clear, right? And then we will move on to single membrane bound organelles. Single membrane bound organelles include, first is Golgi bodies. Okay, just a second, ma. Yeah, first it includes Golgi bodies. Second is endoplasmic reticulum. We have two types of endoplasmic reticulum. One is smooth endoplasmic reticulum. Second is rough endoplasmic reticulum. Fine, right? Smooth and rough endoplasmic reticulum. And then we have lysosomes and vacuoles in an animal cell are very primitive. Okay, remember in a plant cell, vacuole is occupying 90% of the plant cell volume, whereas in an animal cell, vacuoles are very primitive, negligible or absent. Remember that. And then move on to the one more single membrane bound organelle, that is peroxisome, which is highlighted over here. Look into all the labelings properly. Okay, here we go with double membrane bound organelles. First is what? Nucleus is one double membrane bound organelle, then it is mitochondria, after which we have nucleus and mitochondria, only two double membrane bound organelles. Clear till here? Any doubt anywhere, ma? Animal cell is clear, right? Yeah. Good afternoon. Good afternoon, Pradeep. Continue with the class, ma. Animal cell. Okay. Clear till here? Plant cell and animal cell is clear, right? Now we will look after the major differences between a plant cell and animal cell. Plant cell, of course, it is made up of cellulose. Animal cell, cell wall is absent. Shape is regular rectangular shape. Here it is irregular or round shape in an animal cell. Plastids are present. Here plastids are absent. Large central vacuole in a plant cell. Central vacuole or vacuole is temporary or negligible in an animal cell. Centrioles are absent in a plant cell. Centrioles are present in an animal cell. And lysosomes, if you see, lysosomes are rare or absent in a plant cell, and in an animal cell, lysosomes are present in abundance. Nutrition is autotrophic, here it is heterotrophic. Flagella or cilia are very rare, here they are often present. And the cytokinesis, how cell membrane or cell wall division happens? It is through the formation of cell plate from the center to the periphery, which is centrifugal in a plant cell, and it is from periphery to the center by the formation of a cleavage furrow like this, from periphery to the center, which is centripetal in an animal cell. Okay, this is centripetal. Fine, right? This is centripetal and this is centrifugal. Stored food material is starch in a plant cell and glycogen in the animal cell. One more major difference I can tell you is this, the medium of this class is English, ma. Please try to listen and continue, try to understand. If you are not getting anywhere, I will help you. Okay. Now, so here if you see one major difference of Golgi bodies again in between plant cell and animal cell. Golgi bodies. So you can see large interconnected Golgi bodies in an animal cell. Golgi bodies are large and interconnected. You can see interconnected Golgi bodies in an animal cell. Interconnected Golgi bodies in the animal cell, whereas unicisternal or unconnected Golgi bodies you can see it in the plant cell. Okay, unconnected Golgi bodies you can see it in the plant cell like this, okay, which are not connected. This type of Golgi bodies in a plant cell are addressed as dictyosomes. Dictyosomes. This is one difference in between plant cell and animal cell, that is about Golgi bodies. Interconnected Golgi bodies, interconnected cisternae are present in a animal cell, whereas dictyosomes which are unconnected Golgi bodies, these are present in a plant cell. Clear till here? Any doubt anywhere, ma? Yeah. So till here it is fine, right? Differences between a plant cell and animal cell. Now we will move on to the outer limiting boundary of a plant cell, that is cell wall. Okay. First we will see how a cell wall in a plant cell is formed. How a plant cell wall is formed. Let us suppose these two are a plant cells. these two are a plant cells, let us suppose. First and foremost, the outer membrane or the outer layer, first layer which is formed during the time of cell division. Here I will show you. Let us see, this is the telophase, which is the last phase of karyokinesis. Here you see the nuclear genetic material is there at the both ends, and at the center first cell plate is formed. This plate from the center, it will move on to the periphery like this, from center to periphery, from center to periphery. This mode of cell plate formation is called as what? Centrifugal type of cell plate formation or cell wall formation. The cell plate which is formed at the center, as it is moving to the periphery, once it is completed adjoining to the two cells, this is known as what? Middle lamella. First formed layer is middle lamella. Remember, this middle lamella is first formed layer of plant cell wall. Okay. Middle lamella is rich in calcium pectate and magnesium pectate. Fine, right ma? So, the outermost wall layer which is formed in between two plant cells here, the outermost layer which is formed is what? Outermost layer formed is what? Middle lamella. Clear, right? Yeah, let us suppose this is a plant cell with a cell membrane, right? Now, the outer membrane is clear to you, which is composed of calcium pectate and magnesium pectate, and this is the first formed cell layer, right? And next, inner to the middle lamella, once middle lamella is formed, inner to the middle lamella, the second formed layer is primary cell wall. Okay, inner to the middle lamella, primary cell wall is formed. This primary cell wall is meristematic and it is parenchymatous. Okay, primary cell wall is meristematic and it is parenchymatous. Fine, right? Next to the primary cell wall, we have 2 to 3 layers of secondary cell wall. Okay. Next to the primary cell wall, what do we have ma? 2 to 3 layers of secondary cell wall. Fine, right? So, 2 to 3 layers of secondary cell wall. Fine till here. So, you are clear with middle lamella, next primary cell wall and secondary cell wall. Here I have to tell you few things. If you consider a ripening, cell ripening during the time of fruit formation or fruit ripening, where cell also gets ripened, right? So, at this time the middle lamella will get crushed and the calcium pectate, magnesium pectate will get solubilized. That will give you the brittle nature of the fruit. Okay, you can take the examples of sapota or custard apple, anything. Okay, so in the ripening fruits, that brittle nature is because of what? Solubilized calcium pectate and magnesium pectate of middle lamella. Remember that. Okay, and primary cell wall is single layered, which is parenchymatous and it is meristematic, right? And 2 to 3 layers of secondary cell wall. As the cell grows, as the cell wall layers of the secondary cell wall is increasing, secondary cell wall layers as they are increasing, the primary cell wall layer will get crushed. Okay. So, if you take the flow from cell membrane to outer layer, from cell membrane if you see, cell membrane first, followed by what ma? Followed by secondary cell wall, followed by primary cell wall, followed by middle lamella. Okay, this will be the flow from cell membrane to middle lamella. If you take from middle lamella to cell membrane, see, outer is middle lamella, next will be primary cell wall and then will be secondary cell wall and then innermost it will be cell membrane. Clear till here? Now we will look into the composition of a plant cell. This flow chart is very important, ma, and the points that we have discussed for middle lamella, primary cell wall, secondary cell wall, highly important, not just for your EAPCET but also for your NEET. Okay. And then if you see the composition of cell wall, okay, composition of cell wall is first insoluble polysaccharides like cellulose and hemicellulose. See here, you can see insoluble polysaccharides like cellulose, hemicellulose, pectins and proteins. This is the composition of plant cell wall. If you see algal cell wall is composed of galactans, mannans, minerals like calcium carbonate. Okay, it consists of galactans, mannans, minerals like calcium carbonate, algal cell wall. Fungal cell wall, if we see, fungal cell wall is composed of chitin, which is a polymer of the monomer N-acetyl glucosamine units. Here let us remember the two kingdom system of classification which we have dealt in the biological classification. Two kingdom system of classification is proposed by whom? Carolus Linnaeus, where the main criteria of classification is cell wall. Okay, those which consists of cell wall are placed under the plant under the kingdom Plantae despite of their nutrition, right. As such, fungi is also included in the plant kingdom under two kingdom system of classification, right. As such, if you see, fungal cell wall composition is chitin, which is a polymer of N-acetyl glucosamine. Next, algal cell wall composed of galactans, mannans and calcium carbonate. Next, plant cell wall consists of insoluble polysaccharides like cellulose, hemicellulose and pectin. And these are the wall layers from cell membrane to middle lamella and middle lamella to the center, that is cell membrane. Till here it is clear, right? Cell wall is clear, right? So, these are the first formed layer is middle lamella. After middle lamella next it is primary wall, and then secondary wall which is close to the plasma membrane, and next you see the plasma membrane. Clear till here? Okay, clear with the image? And then cell membrane. Okay, so cell membrane is widely studied after the invention of electron microscope in 1950s. Okay, cell membrane or plasma membrane is widely studied after the invention of electron microscope in the year 1950. Here we go. Okay, microscope in the years 1950s. The widely accepted model of a plasma membrane or cell membrane is proposed by Singer and Nicholson. Singer and Nicholson in the year 1972, and the model is what, ma? Fluid mosaic model, right? The model is what? Fluid mosaic model. According to this model is proposed after completely deducing or analyzing the erythrocyte under the erythrocyte, that is RBC. Okay, erythrocyte, that is RBC, under electron microscope. Okay, so here first we will see the composition of cell membrane. Okay, composition of RBC. Composition includes, composition includes carbohydrates, proteins and lipids, right, ma? First major composition is proteins, then lipids and then carbohydrates. Just look into it properly. In a minute I will be back. See, here we go with proteins. First you see, ma, proteins based on the ease of extraction of proteins from the cell membrane. We have three types of proteins. First is peripheral proteins. Okay, first is peripheral proteins. Second is integral proteins. And third is transmembrane proteins. Fine, right? First is peripheral, next is integral and transmembrane proteins. And in RBC, the composition of proteins will be 52% of proteins and lipids are just 40% of lipids are present in the RBC cell membrane. And if you see the carbohydrates, carbohydrates are just 3 to 8%. 3 to 8% of cell membrane will have carbohydrates. Fine, right? And here if you observe the peripheral proteins, they are present at the peripheral ends. They are easily extracted. Okay, they are present at the peripheral ends on the either ends of the cell membrane. Okay, and the integral proteins are embedded inside the cell membrane. Integral proteins will transpan two sides of the cell membrane. Transmembrane proteins will transpan any one side of the cell membrane. If you can see here from the diagram, they will transpan only either sides, only any one side of the cell membrane or both the sides. That is the reason we will say all integral proteins are transmembrane proteins, but not all transmembrane proteins are integral proteins. Okay, all integral proteins are transmembrane proteins, but not all transmembrane proteins are integral proteins. Have a clear note of it. Okay, and if you see, the cell membrane is completely composed of lipid bilayer like this. Fine, right? It has the lipid bilayer. Major composition of lipids in the cell membrane is phospholipids. Cell membrane is highly composed of phospholipids. Lipid will have hydrophilic polar head and hydrophobic non-polar tail. It has hydrophilic polar head and hydrophobic non-polar tail. Hydrophobic non-polar tail. Fine, right? These are the two ends. One is hydrophilic polar head and the next is hydrophobic non-polar tail. This is the lipid composition. So, the quasi-fluid nature, the lipid is just fluid in nature. Remember, the cell membrane, which is what nature ma? Fluid in nature. This quasi-fluid nature of lipids will enable the lateral movement of proteins in the both ends. Remember that statement keenly. We will get in the statement based question. The quasi-fluid nature of lipids, the fluid nature of lipids will enable the lateral movements of proteins in the lipid bilayer. Fine, right? So, this is the complete story of lipids. It is the fluid nature of lipids and how you can see proteins that are present in the lipid bilayer. Proteins are present as iceberg in the sea of lipids. Okay, how proteins are present? Proteins are like, these are two keynote points, please take. Proteins are appearing or present like icebergs in the sea of lipids. And this is the second point that you have to be aware of. That is what ma? Quasi-fluid nature of lipid will enable, this will enable lateral movement of proteins. Fine, right? These are the two keynote points that you have to take. Next is about carbohydrates. Carbohydrates are present in the lipids. Okay. These are in the form of glycolipids and glycoproteins. Fine, right? These are in the form of what? Glycolipids and glycoproteins. Fine, right? They are least in their composition. The major role of carbohydrates in the cell membrane is cell to cell interaction. Okay? This is the complete composition of what? Proteins, lipids and carbohydrates. You can take a screenshot of it, and if you are having any doubts, you can ask me. Ma'am, there are two cell walls. Cell wall layers, beta. I don't know your name, but then we have cell wall layers. We have 3 layers of cell wall. The outermost is middle lamella, which is first formed. Inner to that we have primary cell wall, and next we have secondary cell wall. Fine, right? Secondary cell wall is 2 to 3 layered. Okay. Any doubt anywhere, ma? Yeah. Here we go. About functions of the plasma membrane. Plasma membrane functions, if you see, first it is helpful in the cell growth. Next, formation of intercellular junctions. Formation of intercellular junctions which will help in cell to cell interaction. Fine, right? Next is in secretion. Okay, cell membrane secretion inside the cell, and then endocytosis, that is cell engulfing. Okay, endocytosis, which is also addressed as cell eating. Fine, and then it is also helpful in cell division. These are the major functions of plasma membrane. Next we will see the transport. Cell membrane, which plays a crucial role in the transport. We have 2 types of transport, ma. One is passive transport, second will be active transport. Passive transport and active transport. Passive transport is again of 2 types. One is diffusion, second is facilitated diffusion. This is across the concentration gradient. Passive transport is across the concentration gradient. Fine, right? This is across the concentration gradient. That is along the concentration gradient, where the molecules will move from higher concentration to lower concentration. They will move from high concentration to low concentration like this. Okay, and if you see the active transport, this is against the concentration gradient. Active transport is against the concentration gradient. As it is against the concentration gradient, active transport is ATP dependent or ATP mediated process. Fine. Any doubts still here? So, from low concentration to high concentration, the molecules are transported with the help of ATP. Okay, energy requirement process or energy mediated process, whereas facilitated diffusion and diffusion does not require any energy. Diffusion is for gaseous molecules, it will diffuse directly, whereas facilitated diffusion is for some polar molecules or like water molecules. This will be the movement of the molecules is facilitated either through channel proteins or carrier proteins. Either through channel proteins or carrier proteins. Fine. Any doubts anywhere? Till here everything is clear, right, ma? Cell wall, cell membrane and its functions. Till here it is clear, right? Now we will move on to the single membrane bound organelles. When we have to speak about single membrane bound organelles, first and foremost thing comes into picture is endomembrane system. Endomembrane system includes all the single membrane bound organelles whose functions are coordinated. Okay, single membrane bound organelles whose functions are functions are coordinated. Fine, right, ma? So first you see endoplasmic reticulum. Where do you see this endoplasmic reticulum? Endoplasmic reticulum can be in the form of cisternae or tubules or vesicles. Fine. Cisternae are of these structures. These are cisternae and these are tubules and these will be vesicles. Endoplasmic reticulum can be any of these structures which are extensions of nuclear envelope. Okay, these are extensions of nuclear envelope. Fine, right? Next, this endoplasmic reticulum can be of two types. If they are having ribosomes above these structures, it is addressed as rough endoplasmic reticulum. Despite of ribosomes, if ribosomes are not present, it is known as smooth endoplasmic reticulum. Rough endoplasmic reticulum will synthesize naive proteins. Okay. Rough endoplasmic reticulum will synthesize naive proteins, whereas smooth endoplasmic reticulum will synthesize lipids and sterols in an animal cell. Fine, right? This in the form of vesicles, they will be passed on to the cis phase of Golgi. Okay, they will be passed on to the cis phase of Golgi. Inside Golgi complex, I am briefly explaining you the endoplasmic reticulum. Once you understand this on whole, we will look into each organelle in detail. Okay, so firstly, what you have to understand with respect to endoplasmic reticulum. Endoplasmic reticulum is nothing but these are the extensions of nuclear envelope, and this can be in the form of cisternae, tubules and vesicles. And we have two types of endoplasmic reticulum. One is rough endoplasmic reticulum and second is smooth endoplasmic reticulum. RER will have naive proteins, will synthesize naive proteins. Smooth endoplasmic reticulum will synthesize lipids and sterols. This will be accepted by cis phase of Golgi bodies. Okay, this will be accepted by what? Cis phase of Golgi bodies. And inside Golgi complex, what process will happen? Inside Golgi complex, complete process, packaging and transport of molecules will happen. Okay, so complete process, packaging and transport. This occurs inside the Golgi bodies. Now, from mature phase or trans phase, lysosomes will be secreted out, and these lysosomes are popularly addressed as what? Suicidal bags of the cell. Okay, lysosomes are suicidal bags of the cell. Fine, lysosomes are suicidal bags of the cell. As such, we have primary lysosomes, secondary lysosomes, residual bodies and autophagosomes. We are going to learn all the four types of lysosomes. And vacuole also is included under the endomembrane system if you include a plant cell. If you consider an animal cell, you can eliminate out the vacuole because vacuoles are primitive or negligible in an animal cell. Vacuole is the last organelle of endomembrane system wherein the residual bodies or the digested toxins, the toxins which are present in the cytoplasm are sent from the cytoplasm to the vacuole, from the cytoplasm to the vacuole through, from cytoplasm to the vacuole through active transport. As the pH inside the vacuole is less and it is more in the cytoplasm, it is transported in what? Active transport. Fine, right? and this is the last organelle that we are going to look into. Clear with the complete picture ma? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuole. If you are brief and clear on this note, it is fine. If you are still having any doubt, you just ask me, I will clarify it. Now, we will move on to endoplasmic reticulum. As I told you, endoplasmic reticulum can be of what structures ma? Cisternae, tubules and vesicles. Mostly rough endoplasmic reticulum which you are seeing here is in the form of cisternae. You can see it here clearly. Smooth endoplasmic reticulum will be mostly in the form of vesicles and tubules. Clear, right? So, here how ribosomes are connected to rough endoplasmic reticulum. The ribosomes are adjoined by a protein called as ribofourin. Ribofourin. This ribofourin will attach the larger subunit of ribosomes, that is 60S, and smaller subunit 40S. The larger subunit is connected to the ribofourin which adjoins the ribosome to the endoplasmic reticulum. So, this is what type? 80S type of ribosomes. Okay. So, clear? This is about the rough endoplasmic reticulum. As I told you, RER will be synthesizing what ma? Naive proteins. These proteins are synthesized in the form of vesicles only. Okay. And smooth endoplasmic reticulum will be synthesizing lipids, and in an animal cell this will also help in the synthesis of steroids. Animal cell, it will help in what? Steroid synthesis or sterol synthesis. It will regulate the calcium ion concentration. Calcium ion concentration is regulated. Fine, right? Calcium ion concentration is regulated, so that this will help in the muscle contraction and relaxation. Okay? Calcium ion concentration is regulated by SER, which will help in muscle contraction and relaxation. Fine. Any doubt still here? Everything is clear, right? Yeah. Next you see Golgi bodies. In Golgi bodies, if you analyze it properly, the cis phase of Golgi is towards the endoplasmic reticulum. Cis phase of Golgi is convex or forming phase. Okay. This is also known as cis phase, convex phase or convex side or forming phase, which will take all the vesicles, both lipids and proteins will be collected here, and inside the Golgi bodies, okay, inside the Golgi bodies, first of all Golgi bodies are having what? Cisternae. Okay, these cisternae are interconnected. Inside Golgi, packaging will happen. Okay, packaging and synthesis of glycolipids, glycoproteins. Synthesis of glycolipids and glycoproteins will occur inside the Golgi. Okay, glycolipids and glycoproteins from mature phase or trans phase. Okay, this phase is known as, the later phase is mature phase or trans phase of Golgi, which is also known as concave side. Fine. from this end all the vesicles will be released out. This can be intracellular targets or extracellular targets, okay, which are intracellular and extracellular targets. Fine, right? Intracellular and extracellular targets will be released out from the trans phase of Golgi bodies. And glycolipids, glycoproteins are also synthesized inside the Golgi bodies. Now we will look on to in which type of cells Golgi bodies are abundantly present, in which type of cells Golgi bodies are absent. So here we go beta. First thing, yeah, Golgi bodies are abundantly present. They are abundantly present. Take down, very important. They are abundantly present in root cap of a root, wherein this will promote positive geotrophic movement. As Golgi bodies will synthesize glycolipids and glycoproteins, they will help in lubrication and they will promote positive geotrophic movement, okay, positive geotrophic movement. And the next is they are abundantly present in acrosome of a sperm cell. Now we will see in which type of cells it is absent. Golgi bodies are absent in, if you see, first is sperm cells of bryophytes and pteridophytes. And these are absent in mammalian RBC cells, they are absent in mature sieve tube cells. Clear till here? Any doubt anywhere, ma? Fine, right? Take down these points, very important, not just for EAPCET but also for NEET. Take down it more keenly. Here we go with next organelle, that is lysosomes. So the vesicles which are synthesized from the mature phase of Golgi bodies, these are also addressed as Golgian vesicles. Golgian vesicles. These Golgian vesicles will be synthesized out. These vesicles which are rich in hydrolytic enzymes are first addressed as primary lysosomes. Okay. So the primary lysosomes are rich in what, ma? Hydrolytic enzymes. First type of ribosomes is primary lysosomes. Primary lysosomes are rich in hydrolytic enzymes. These hydrolytic enzymes are inactive. Hydrolytic enzymes are inactive. Okay. Primary lysosomes will have what, ma? Hydrolytic enzymes. Hydrolytic enzymes, for examples if you take, okay, for example, carbohydrases, carbohydrases, proteases, lipases and hydrolases. These are the examples of what? Hydrolytic enzymes. And next is, second is secondary lysosomes. Secondary lysosomes. These are also known as heterophagolysosomes or digestive vacuoles. Heterophagosomes. Okay, or digestive vacuoles or digestive lysosomes also we can call, because in this secondary lysosomes, these are nothing but primary lysosome along with a digestive vacuole or a molecule you can say. Okay. So, after digesting the digestive molecules which are present, okay, or the food particles, you can say it clearly, okay, primary lysosomes along with the food particles that are present inside the cell. Okay. This will release out some molecules, right. These are addressed as what? Residual bodies. Okay. So, the third is residual bodies. After digestion, few particles will be sent out inside the cell, right. These are known as what? Residual bodies. And after residual bodies, third is what? Autophagosomes. Autophagosomes. Autophagosomes are nothing but primary lysosomes along with dead particles or dead substitutes that are present inside the cell. Dead particles or dead substitutes that are present inside the cell are known as what? Autophagosomes. Clear, right? So, we have 4 types of lysosomes. Firstly, lysosomes are addressed as suicidal bags of the cell, because inside the cell, whichever digestive molecules are present or dead particles are present, those are digested by what? Lysosomes, as lysosomes are rich in hydrolytic enzymes. So, primary lysosomes along with food particles which are present inside the cell are addressed as secondary lysosomes or heterophagosomes. They will be releasing out some particles after digestion which are known as what? Residual bodies. And if primary lysosomes are adjoined with some dead particles inside the cell, those are known as what, ma? Autophagosomes. Till here everything is clear, right? Now, last is vacuoles. Last is vacuoles. Here we go with vacuoles and end of the session. As we have discussed, in a plant cell, vacuole is of how much percent, ma? Vacuole occupies 90% of plant cell volume. Fine, right? And vacuole is surrounded by a membrane called as what? Tonoplast. Here what happens is, vacuole consists, the pH of vacuole will be very less. So, it is acidic. Whereas, pH of cytoplasm, if you consider this as the cytoplasm pH will be more. So, compared to the vacuole, the pH is basic. Cytoplasm pH is more compared to the vacuole. So, from cytoplasm through active transport, the molecules will be transported, all the toxins that are being digested after the lysosomes, after lysosome digestion, be it with respect to dead particles or with respect to food particles, all the toxins which are present in the cytoplasm are sent to the vacuole by what transport beta? By active transport. Okay, this is by active transport. This is the complete sum up of what? Vacuoles. Clear till here, right? So, at the end you have to remember this major point, that endomembrane system includes 4 organelles: endoplasmic reticulum, Golgi bodies, lysosomes and vacuole. And with respect to each organelle, we have dealt it in more clear way. Fine, right? If you are having any doubts till here, you can ask me. In our next session, we will be looking up to centrioles and ribosomes, which are non-membrane bound organelles, and we will look into the cytoskeleton structures, microtubules, microfilaments and intermediary filaments, and we will also sum up the double membrane bound organelles, which are very important. Okay, so by tomorrow we will finish off this chapter and we will start cell cycle and cell division as well. Okay, so that's all for today's session beta. Make use of these live sessions where complete content along with EAPCET previous year questionnaire practice will be given to all the students. So make use of this golden opportunity and finish off your concepts as such. And with regards to preparation or anything with regards to the subject, you can drop a message in the chat box where I will be addressing your doubts in person. Okay, so that's all for today's session. Thank you.